Alright, so today we're going to be talking about applied genetics, and here in a few minutes we're going to be talking about applied DNA technology. They kind of go hand in hand. So the first thing that we're going to talk about with applied genetics is going to be selective breeding. So for thousands of years, humans have chosen plants and animals with certain desired characteristics that they particularly needed in a certain situation, and then bred them together. So what they wanted to accomplish with this is that they wanted to increase the chance of that characteristic that they desired. They wanted to increase the chance of that showing up in the population. So the process of selective breeding requires several generations of offspring before your desired trait will become common. If you take a desired trait, say maybe you have a tall corn stalk that you want to continue to grow tall, well, first you have to find two tall corn stalks, breed them together, but your offspring from that generation, it could vary depending upon all the different types of traits or the different types of genes that those corn stalks may have. So it will take several different generations and several generations down the line for you to start to see a real improvement or a real more widespread occurrence of that particular trait that you're looking for. But one example that we have is milk production in cattle. So this is over a course of 50 years, so again, several generations later. In 1947, an average milk cow would have produced about 5,000 pounds or 4,997 pounds of milk per year. But 50 years later, in 1997, after the farmers had continuously bred milk-producing cows, or cows that produce a lot of milk together, in 1997, they produced 16,915 pounds of milk. So over 3 times the amount that the original cow back in 1947 produced. So your VIC number 1 is selective breeding increases desired traits. So anything that you're looking for in a desired trait could be something from a plant, could be something from an animal, anything that you desire in a specific organism. Selective, if you take that organism and you breed it with another that is also expressing that same trait to try and get more of that trait, that is your selective breeding. Alright, so breeders will often use the method of inbreeding to eliminate undesired traits. So inbreeding is the mating of two closely related individuals. Now these may be brother and sister, they may be even first cousins, but normally that's going to be about as close as you get. You can sometimes have mother and daughter, or mother and son, father and daughter, things like that. But remember, these are going to be with things like offspring from plants or animals or things like that. But, however, we might be familiar with this, inbreeding can also bring out harmful recessive traits. The reason for that is because two organisms that are closely related are going to have the more likely chance that they might have the same harmful recessive trait. So you might see some recessive traits popping up in the inbreeding or in the organisms and in the offspring. However, the end result after many generations of breeding may result in a new breed. So for instance, if you've ever heard of the example of a Labradoodle, whenever they breed Labradors and Poodles together, after several generations of doing that, they've kind of formed this new type of dog that they consider its own breed. So a breed is a selected group of organisms within a species. So for our circumstance, or our example, would be a dog that has been bred for particular characteristics. So the Labradoodle, their coat color depends just like a Labrador's will, but they're kind of have the kind of hair characteristics of a poodle, and they're a lot bigger. So that particular breed, those are the particular characteristics that they were looking for in that Labradoodle breed, but it is still part of the dog species. So hybrids, remember, those are just heterozygous for traits, are usually bigger and better than their parents. So if you have two homozygous parents, maybe they're both particularly tall, but if for some reason you have a heterozygous offspring, normally those are going to be bigger, stronger than their parents. Alright, so how do we determine genotypes? It's impossible to know whether an organism is heterozygous or homozygous dominant, so whether or not they have a dominant and recessive allele or just two dominant alleles, by looking at it. You cannot tell whether or not, say, a cornstalk, if it's tall, if it is homozygous for tall traits, or if it's heterozygous for tall traits. For this reason, breeders must perform what is called a test cross, and they can do this both with plants and with animals. So a test cross is a cross of an individual with an unknown genotype that we want to know, with an individual of a known genotype. So they take an organism that they know what the genotype is, and they cross it with one that they don't know what the genotype is. The pattern of phenotypes, which remember, phenotypes are your physical characteristics in the offspring, can help determine the unknown genotype of the parent. So usually what you're going to see is the parent with the known genotype is going to be homozygous recessive for the trait. If they are homozygous recessive, that means that your recessive trait has to be shown. It's pretty easy to determine if an organism is homozygous recessive. Again, that recessive trait has to be shown, so that cornstalk might be particularly short, which would be your homozygous recessive. All right, so now we're going to talk about applications of DNA technology. So scientists, they take these different genotypes and these different things that they've already looked at from their applied genetics, but they're also particularly interested in cloning entire animals. So the most famous that we have cloned so far is Dolly the sheep in 1997 in a farm in Scotland. And the cloning of animals would allow farmers to increase productivity. So, for instance, they might be able to take a cow that produces a lot of milk, not have to worry about that selective breeding process, just be able to directly clone that cow so that that next cow that they have would produce the same amount of milk as the original cow. So kind of cutting out the middleman and cutting out the many generations of offspring that you would have to rely on elsewise. Now, they also rely on DNA for sequencing DNA. So, for instance, the Human Genome Project. Genetic engineering allows scientists to produce pure DNA that they can look at under a microscope and be able to tell exactly what sequence of our correct sequence of our DNA bases would be. So, how are these bases paired, and in what sequence are they paired, and in what way might we experience a mutation or something wrong with our DNA? Now, recombinant DNA, which we looked at, that was our lecture for yesterday, is used to produce chemical compounds that are used by humans. So your example, they use many of these different compounds for use by humans, but your first example, however, will be the bacterium E. coli, which can cause, the sickness E. coli, which can cause food poisoning, can be modified to actually produce the indigo dye that they use to color blue jeans. So that bacteria, they can actually use that to produce a dye that we use to color our clothes. So we can use a lot of these chemical compounds. All right, now pharmaceutical companies use recombinant DNA to treat human diseases. So one of the examples that we can look at is the human gene for insulin is inserted into a bacterial plasmid, and remember we talked about plasmids with our recombinant DNA, to replicate and produce insulin for diabetics. So this is one of the ways that diabetic people can actually get their insulin that they need, that their body is incapable of producing, by this bacterium, and then they inject themselves with the insulin that they receive from the recombinant DNA. Now, scientists can use transgenic animals to study specific diseases and the role that genes play in an organism. So, for instance, if a specific organism may be missing a gene, how will that affect the organism? So, example, a transgenic sheep was produced that, what this should say, or accidentally erased it, would say, contain the corrected gene for hemophilia. So if you'll remember from our study of hemophilia, hemophilia is whenever a person is lacking the protein that will cause their blood to clot correctly. So they will be what is called a free bleeder and continue to bleed until they receive medical attention. Now, where the DNA was placed in this sheep, that actually allowed for the production of the blood clotting protein to form in the sheep's milk. So people were able to take that protein, scientists were able to take that protein directly out of the sheep's milk and use it to help people that had hemophilia. They were able to get injections and transfusions of that blood clotting protein, and then they were treated for at least a little while. Now, your Vic number 2, recombinant DNA has many practical applications, but there are several moral and ethical questions surrounding its use. All right, so in agriculture, how do we use recombinant DNA in agriculture every day? Well, one of the ways is that crops have actually been developed to taste better, so like corn, for instance, stay fresher longer so that it's able to be shipped farther away to areas that maybe, say if it came from California, well, by the time it gets to North Carolina several years ago, it might have been rotten. But now, thanks to recombinant DNA, they can actually cause it to stay fresher longer. They can actually cause them to be protected from disease and from insect infestations, things that make them resistant to these type of infestations and these type of diseases, using that recombinant DNA. So currently, we actually have a lot of research being done to increase the vitamin content in crops. And this has a particularly special application in that this can help to supplement the diets of those in underdeveloped countries. A lot of people in underdeveloped countries don't really actually get the type of nutrition that they need. This is a pretty well-known fact. But what we're working on now is increasing the vitamin content in things like corn, things that are cheap to produce, so that we could possibly donate that corn to somewhere that is underdeveloped, somewhere to help supplement their diets and supplement their nutrition so that they're actually getting those nutritional values and that nutritional needs that they have. Now, these organisms, either be plant or animal, are referred to as genetically modified organisms, or GMOs, and are the source of much controversy surrounding a lot of different types of debates currently in the political scene.